Hi to everyone. It's Margherita Rimini, and I am from Milan, from the San Raffaele hospital. I will talk to you about our works, which is focus on the clinical impact of brcaness phenotype in advanced cholangiocarcinoma patients. The concept of brcaness has been proposed to indicate the phenotype that the sporadic tumors share with familial BRCA cancers, thus concerning not only BRCA1 or 2 alterations, but also abnormalities in modifying the proteins which regulate genomic aberration and which are involved in gene expression, resulting in the distinctive DNA repair defect due to homologous recombination deficiency. The brcaness phenotype, more precisely the presence of BRCA1 or 2 mutations, have been highlighted to have a clinical and therapeutic impact in a number of cancer settings, including breast cancer, ovarian cancer, prostate cancer, and interestingly, in pancreatic cancer. More specifically, cancer and with BRCA mutation and HRD are considered to have a specific susceptibility to drugs which induce breaks in DNA, including platinum-based chemotherapy and PARP inhibitors. The role of HRD alteration in cholangiocarcinoma is still widely unknown, and only few data about the clinical impact are already available. The aim of the present work was to investigate the potential impact of brcaness phenotype in response to platinum-based chemotherapy in advanced cholangiocarcinoma patients. We saw retrospectively identified 150 consecutive patients treated with platinum-based chemotherapy for unresectable or metastatic intrahepatic cholangiocarcinoma cancer from 5 Italian institutions and 1 Spanish institution. A full histopathologic review was performed by an expert gastrointestinal pathologist, and the whole genome analysis of the primary tumors was performed by the Foundation 1 SAE. The definition of brcaness phenotype in cholangiocarcinoma has been translated from the advanced pancreatic setting, as you can see in this semi-dot box. In the left side of the results box, you can observe the clinical and molecular characteristics of the sample at the baseline. Overall, 150 patients have been included in the study, and 72 were brcaness mutated and 78 were brcaness wild type. In the first table, you can see the clinical characteristics of patients at the baseline. As you can see, the two groups of patients were almost homogeneous, except for the proportion of male versus female and for the number of patients resected. Of interest, a large proportion of patients in both the groups was diagnosed with metastatic disease in liver and nose, and were treated with more than 2 lines of systemic treatment for advanced disease. In the diagram above, you can see the most common mutated genes in the sample, which were CDKN2A, ARID1A, CDKN2B, KRAS and RAS, BAP1, BBRM1, and TP53. Despite the different distribution of gene mutations between the two groups of patients, after excluding all genes included in the definition of brcaness, no statistical differences were detected between brcaness mutated and wild-type patients, except for the gene CRABBP, which was highlighted to be mutated exclusively in the brcaness mutated group of patients. In the right side of the results box, you can see the survival analysis. For the 150 patients who received chemotherapy for unresectable, for metastatic, or for recurrent disease, correlative analysis between the genetic alteration involved in brcaness phenotype and the survival outcomes were performed. At the univariate analysis, brcaness was associated with a longer progression-free survival, and a trend toward a benefit in survival was found, which did not reach the statistical significance. Moreover, patients with a brcaness mutation show a higher proportion of a disease control rate. Following adjustment for a clinical recovery as no as prognostic factors in this setting, including ECOG performance status and stage, the multivariate analysis confirmed the brcaness as independent favorable prognostic factor for PFS, and a positive trend was found for OS. At the best of our knowledge, the present work is one of the first evidence of an increased benefit from platinum-based chemotherapy in patients with cholangiocarcinoma harboring a brcaness phenotype compared to those without. I would like to focus the attention on the overall survival curves, which show two trends. In the first part, the two curves split up in favor of a patient with a brcaness mutation, but after progression to platinum-based chemotherapy, the two curves experience a cross. In other words, this data seems to suggest that brcaness mutated patients may have a benefit from a platinum treatment, but that once the C maximum C benefit has been achieved, a maintenance treatment able to freeze this response could be a good option. And starting from the biological similarities with the C pancreatic cancer, our analysis opened the question of whether even patients with cholangiocarcinoma with brcaness phenotype could benefit from a maintenance treatment with PARP inhibitor after response to platinum salves. Indeed, in the last table, you can see the clinical outcomes of 3 patients included in the sample, which were treated with PARP inhibitor. All these 3 patients were diagnosed with a locally advanced disease and received radical surgery at the diagnosis. Then, all these patients received systemic treatment following the diagnosis of recurrent disease. Unfortunately, both patient 1 and 3 had to stop the treatment early due to adverse events. On the other hand, the patient 2 carrying the mutation in ATM experienced a PFS of 10.3 months and OS from the start of treatment of 1.4 months and an OS from the diagnosis of 55.3 months. In conclusion, despite the retrospective nature, the present analysis performed on a significant cohort of cholangiocarcinoma patients demonstrated a positive prognostic impact of brcaness phenotype on PFS, and a trend toward a better OS was observed. This data presented may suggest future investigation on the molecular landscape of brcaness cholangiocarcinoma patients, as well as prospective trials on larger cohorts to validate the predictive role of brcaness phenotype in advanced cholangiocarcinoma patients receiving platinum-based chemotherapy, eventually followed by a maintenance treatment with PARP inhibitors.